Now we are going to do this activity to observe that chlorophyll is present in leaves. As you know, students, chlorophyll is the internal factor that is necessary for photosynthesis because it's already present in the leaves of green plants. For this observation, we need to use the aquatic plant hydrilla. So here you can see I have a small part of the hydrilla plant. From that, I have put it in water so that the plant is still alive, and I will take a small part of a few leaves from this hydrilla plant, and here I have a clean glass slide. On this, the lesser the leaves, it's better, so I'll put only a few leaves, and also to keep it hydrated and to provide enough water, I will put a few drops of water there. And as you know, without trapping air bubbles, we will have to close it with a clean cover slip. So here I have the clean cover slip and I have prepared the slide containing hydrilla plant. Now for us to observe it under the light microscope. I can place the slide on the stage and make sure there is enough light there. I will set it to low power and also move the slide around so that there is enough light falling on the leaf. Now when we observe it through the low power, you can see the green color chlorophyll or chloroplast present in the cells of the leaf. Here you can see the greenish color cells with the chloroplast in them. But this is not enough to observe it clearly because we will need a higher power to observe the movement of the chloroplast. So I will move it on to higher power. Now you can observe clearly the cells, that is the chloroplast present in the cell. Here you can only observe the chloroplast. But if I take this microscope to a place where there is strong direct sunlight, then since the plant is still alive, you can see the chloroplast moving towards the sunlight, towards the direction of the sunlight. But here, since it's inside the lab, we don't have enough direct sunlight, so you can only observe the chloroplast present in leaves. So students, now through this observation, you have seen that there is chloroplast in leaves, and that is the internal factor that is necessary for photosynthesis. Now we are going to test for the presence of starch in leaves. As you know, students, starch is formed after photosynthesis takes place in the leaf. You know the main product of photosynthesis is glucose. And the glucose gets stored temporarily in the leaf in the form of starch. So if there is starch present in the leaf, that means photosynthesis has taken place. We have discussed this before. So now we will do the activity to test for the presence of starch. For that, I have taken a leaf, a shoe flower leaf. You can take any leaf like this, where you can easily observe the color change after we carry out the procedure. So you know the first step, students. The leaf is not permeable, so we have to boil it in water. So for that, I have a beaker containing water ready. Into this, I will have to put this leaf. I will put the leaf in. I have selected a leaf that can fit into this beaker so that it is easy for us to boil it and take it out. So I have the burner ready and now I have kept it to boil. you will have to observe the changes that are going to take place here. Initially, you saw that the leaf is green in color. So students, now you can see when the water is boiling, you can see a change in the color of the leaf. Earlier it was green in color, now it is turning to a pale brown colored leaf. So we will have to wait for some more time until it really becomes pale in color. And you know the reason for doing this first step. The leaf, the cells are not permeable. So by boiling it, we increase the permeability of the cell by damaging the cell wall and the cell membrane. As you can see students, the water is boiling and the leaf is getting boiled, so the permeability is increasing. Okay students, now that the leaf is boiled enough, I will take it out of water. First I will remove the leaf and place it on this, and also I will need some more water because I am going to do the next step where I will need a water bath. So to this I will add more water. You all know students, the next step we have to boil this leaf in alcohol, and this is the ethyl alcohol, which is highly flammable. It can vaporize quickly and catch fire quickly. Therefore, it is not correct to hold this tube to the direct flame. Therefore, I have to use this water bath. So first I will transfer the leaf into the boiling tube and I will add enough alcohol to cover this leaf so that I don't put excess alcohol. I can prevent vaporization and alcohol catching flame. I will add some alcohol enough to cover the leaf. Here, if I add a small amount, that should be sufficient. Now, I will make sure that the leaf is completely immersed in alcohol. You can see I have put sufficient alcohol to boil the leaf. Here, we will place this inside the water bath. You all know the reason for this step, students. When we boil the leaf in alcohol, now that the permeability of the cells have increased, the chlorophyll will come out of the leaf and dissolve in alcohol. You can see the color change there. The alcohol solution is turning green in color. Earlier, when we boiled the leaf in water, there was no color change. The color of water remained the same. It was colorless. But here, alcohol is turning green in color. That means the chlorophyll is dissolving in alcohol. Okay, students, I think that is more than enough boiling in alcohol. The alcohol has become dark green in color. So, most probably, all the chlorophyll or most of the chlorophyll will be dissolved in alcohol. So, I will remove it from the water bath. I have to place it, let it to cool a little bit. What is the next step? We have to wash this leaf in water. So, for that again, I will take a little amount of water into the beaker and I will transfer this leaf I can take it using the glass rod. Here you can see the alcohol is completely green in color. That means the chlorophyll has dissolved in alcohol. Now I make sure that the leaf is washed completely. Now I can touch it with my hand, no problem. So I will keep the glass rod aside and make sure that it has dissolved and got washed completely. Here you can see we have to make sure that the leaf does not get damaged. I will place it back in the petri dish. So now the leaf is ready for testing. So to see the color change clearly, I will transfer the leaf onto this piece of sponge. So now you can see the color of the leaf clearly, and you can see this region where the color is very light. That means most of the chlorophyll has been removed from that region. Now using the glass tube, I will put a small amount of iodine onto the leaf. As we put it, unlike the starch test where you see the immediate color change, here you will not get the immediate color change. We will leave it for some time, then you will be able to see the color change. I will add a little bit more iodine to the other parts as well. We will have to leave it until the color change takes place. So now you can see the color change, students. In different parts of the leaf, you can see the dark bluish black color or the dark purple color that should form when starch and iodine are present. So this confirms that there is starch in the leaf. So that means photosynthesis has already taken place. So if you can remember, students, we took the leaf from a plant that was exposed to direct sunlight for few hours, and photosynthesis takes place in the presence of sunlight, and therefore we can see the color change with iodine, the dark purple or dark bluish black color formed when there is starch. So with this, we can confirm that photosynthesis has taken place. As I explained to you all, we are going to test whether light is necessary for photosynthesis. So for that, we have a potted plant. I kept this in the dark for 48 hours. So I took it out of the dark now, and I am going to identify two leaves that are similar in nature, so that we can cover one leaf with black color paper and the other leaf with transparent polythene paper. So in order to do that easily, I have already taken two bags, sealable bags, so that it's easy to cover the leaves. One is in black color polythene, covered with black color polythene, and the other one is transparent. So then we provide identical environment except the factor sunlight. Now in this plant, if we look at the leaves, you can see this leaf on this side and the other leaf, these two leaves are all similar in size. So I will take two leaves that are opposite to each other, and then to one leaf I will cover it with this black color polythene bag, but it is a sealable bag, so it's easy for me to cover it completely. In this activity, students, you can also cover part of the leaf with black color tape. you have the black colored tape, so you can paste it around the leaf. You can select an area and paste it as well. Otherwise, we can cover the full leaf with black bag. So, with this other polythene bag, I will be covering an identical, we can't really say identical, a leaf similar to the first one that was covered with black polythene. So, here, so now students, you can see the two leaves covered, one with transparent polythene bag and the other one with black color polythene bag. So, there is no sunlight for this leaf and there is sunlight falling on this leaf. Apart from that, all the factors are same. Now, I am going to take this pot and place it in a place where there is enough direct sunlight. And I'll leave it there for about 2 to 3 hours. Then we will do the test for starch. So, we prepared 2 leaves, one covered with black color polythene, the other one covered with transparent polythene sealable bags. And then I placed the plants in direct sunlight for about 3 hours. After that, I have prepared the leaf for y'all so that it's ready to test for the presence of starch. Y'all know the steps of this procedure. We have to boil the leaf in water first to make it more permeable. Then we boil it in alcohol using a water bath so that the chlorophyll will dissolve in alcohol. And since alcohol is highly flammable, we have to use the water bath. After that, when the alcohol solution becomes green in color, we stop boiling it and then we remove the leaf and wash it in water. Wash it well in water. After that, I have the 2 leaves here. Now, this is the leaf that was covered in transparent polythene. And this is the leaf that was covered with black color polythene. Now, to both the leaves, I will put iodine. If starch is present, then you will see a color change. You know the color change. It has to become dark purple or dark blue. If there is no starch, then there will be no color change. And the presence of starch indicates that photosynthesis has taken place. So, I put iodine to this leaf. I put enough iodine and the same way I'll put iodine to the other leaf. You can already see the color change taking place. So, here you can see, students, the leaf that was covered with black polythene, the leaf that was covered with transparent polythene. And you can see that here there is no color change. The purple or dark blue color has not appeared. But here you can see there is the appearance of dark blue or purple color. That means in this leaf there has been photosynthesis. And this was covered with transparent polythene. So, light is needed for photosynthesis. The second leaf was covered with black polythene, no light, no photosynthesis. Therefore, you don't see a color change that has to be there if starch is present. So, from this activity, we can confirm what? That light is needed for photosynthesis. Okay, students, you know the activity. We are going to test whether carbon dioxide is needed for photosynthesis. So, the materials that we need, I have them here. A potted plant, it's a shoe flower plant, and then have the 2 solutions. One is water, that is labeled as C, you can see here, and KOH solution is labeled as solution D. The same time, I have 2 bags of equal size. They are transparent. So, enough sunlight can penetrate to the leaves, and that is also labeled as C and D. In addition to that, I have written the liquids or the solution that we are going to use. Water in bag C and KOH in bag D. And to tie the bags to the leaves, I have the twines. So, here from the plant, we will have to select two similar leaves. And this plant was kept in the dark for 48 hours. After that, I have brought it here, and now I'll take, you can see these two leaves are somewhat similar, size and opposite to each other. So, the maturity is also the same. So, I will select those two bags. I am selecting these two leaves. So, I will transfer the water into bag C and insert the leaf into the bag. And then we will have to tie it. Similarly, to the other leaf, I will add the KOH solution into the bag, insert the leaf, so here students, you can see one bag contains water, the other bag contains KOH, and this is leaf C, this is leaf D. Now we have to keep this potted plant in direct sunlight for about 3 to 4 hours. I will do that first. So now after keeping the plant in sunlight for about 4 hours, I brought the plant back to the lab. Then what do we do before we test for the appearance of a color change? We have to prepare the leaves. For that, we have to boil the leaves in water first. Then using a water bath, we boil the leaf in alcohol. Why do we use a water bath? Because alcohol is highly flammable. So, to prevent it catching fire, we use a water bath. Then we wash the leaves in water, and now the leaves are ready for the final testing. So, here I have the two leaves, the leaf C. In C we had water, and leaf D, inside bag D we had KOH solution. And I have the reagent that I am going to use, it's the iodine solution. So, what we will do is we will put few drops of iodine onto both the leaves. I'll put it to leaf C first. You can observe the color change, and also to leaf D. It might take a little time. You can observe the two leaves. I will put a few more drops to both the leaves. So students, after I put few drops of iodine solutions to both the leaves, leaf C and leaf D, you can see the observation. You can see in leaf C there is a color change, appearance of purple or dark blue color. But in leaf D there is no color change. There is no purple color or dark blue color. So what can you infer from this? In leaf C there has been photosynthesis. So starch is produced and it gives the color change with iodine. But in leaf D there is no starch, that is why there is no color change. That means there has been no photosynthesis in leaf D. Now if you know the solutions. C was water and D was KOH. So, water does not absorb carbon dioxide, therefore, there is carbon dioxide for leaf C and photosynthesis takes place there. But for leaf D, there was no carbon dioxide. Why? KOH absorbed the carbon dioxide that was there inside that polythene bag. So, there was no photosynthesis in leaf D. So, from these two observations, we can confirm that carbon dioxide is necessary for photosynthesis. Okay, students. So, you know the activity. We are going to demonstrate that chlorophyll, that is an internal factor needed for photosynthesis. For that, we have to use a mosaic leaf. I have few examples of mosaic leaves here. You can see in this leaf, these parts where you can see the yellow color regions. These are the places where there is no chlorophyll. Similarly, take this leaf. Here also in the middle and like a pattern, there is no chlorophyll. Another type of leaf is this one. This also you can see the green color regions and the off white or pinkish color regions that do not have the chlorophyll. So, I am going to use this leaf for the activity today. What I have done is already I have drawn the shape and of the leaf and I have marked the regions. Here you can see if I place it here, it fits exactly with the leaf, and also you can see the regions that are shaded in the diagram is the region that has chlorophyll. So, here you don't have to keep the plant in the dark for 48 hours. Directly I pluck this leaf from the plant. Now, I have to prepare the leaf. You know the steps, students. Boil it in water, boil it in alcohol using a water bath, and then we have to test it for the presence of starch. So, now I am going to do that procedure. So, I have the leaf after preparing it for the test for starch. So, to this we will add a few drops of iodine. Now, I will add iodine to the full leaf so that you can observe the color change. We will have to wait for some time. Okay. Here you can observe, students, in the regions where there is the green color, that is chlorophyll is present, you can see the appearance of dark blue or purple color. But the regions where the leaf is completely not green in color, there is no color change. So, from this observation, we can confirm that chlorophyll is necessary for photosynthesis. Okay, students. So, now I have all the materials that needed to demonstrate that oxygen is evolved during photosynthesis. You all know glucose is the main product and oxygen is the byproduct. So, I have a large beaker with water in it, and here I have an aquatic plant. You can take either Vallisneria or Hydrilla. I have here, this is Hydrilla plant. Then a funnel and additional water and also a test tube to collect the oxygen gas that will be produced during photosynthesis. So, I will first prepare the setup. I will transfer the Hydrilla plants into the funnel. With that, if we can insert it into water, but I have to make sure all the plants are in water. Yes. So, then I will need this test tube. There is little space. So, what I will do is I will add some more water, fill it up to the mouth. Before doing that, I have to increase the amount of water inside this so that there is no air or any form of gas inside the test tube. So, if we fill the water above this level, and I will insert this, and then there is still gas bubbles, so I'll have to add more water. In this activity, students, we have to make sure that there is no air trapped inside the test tube. Now we can invert it inside water so that there will be no air bubbles inside the beaker. Now with this setup, in order for us to be, I will remove a little bit of water from this beaker so that we can easily carry it to a place where there is enough sunlight. So carefully holding it, I will remove a little amount of water. Now you can see, students, the setup is ready. The hydrilla plant is inside the funnel and the test tube contains water only. There are no gas bubbles inside. Now we will have to place this setup in a place where there is enough sunlight or where there is direct sunlight. That is for about 2 to 3 hours. Then afterwards, when we observe, we can see the observations that need to be there. Okay, students. Here I have another setup that I prepared in the same manner, kept it in the sunlight for about 3 hours. Now here you can observe inside the funnel. If you look closely, you can see there are a lot of air bubbles. And also you can see that the water level has decreased a little bit, and on the sides of the test tube also you can see the air bubbles or the gas bubbles actually. So if we move it a little bit, you may be able to see the movement of the gas bubbles. You can see the gas bubbles coming up. So this is how the gas will evolve and get collected in the test tube. So you have to remember, students, there will be observations. One is the evolving of the gas bubbles, you can observe that, and also the water level will decrease and the gas that you already know is oxygen will be collected on the surface of water. So that is above water. Water will be displaced and oxygen will be collected in it. If we still keep this setup in sunlight for some more time, there will be more gas collected. That is, there will be more oxygen collected in it. So now I will keep this in the sunlight and then we will observe it and test to check whether the gas collected is oxygen. Okay, students. Now I left this setup in the sunlight for few more hours. You can see the water level has decreased further and the oxygen gas is collected inside the test tube. So the next step, we will confirm that the gas evolved is oxygen. What do? We have to test it with a burning splint. So to do that, I will have to take out the test tube, making sure that the gas does not escape. So I will have to tilt it a little bit and then you can see there are more gas bubbles coming out. Okay, students, now I will invert the test tube so that the air is on top. You can see it, and now I will insert the glowing splinter. You can see it glowing more. So that is because there is oxygen in it. So students, you saw that this was the normal splinter. Without, you couldn't see the glowing end. But when we insert it into the test tube with oxygen, you saw a better glow. If there was enough oxygen, a very large amount, you can see a flame. Because there was only a small amount of oxygen, you were able to see the glow only. So with that, we can confirm that the gas collected inside the test tube was oxygen. And we can also confirm that oxygen is evolved during photosynthesis. That is the byproduct of photosynthesis.